Hello and welcome to the Digital Plant Health video channel. My name is Alexey Mikaberidze, and in this video I will present a very brief summary of this scientific paper published in Plant Pathology in 2019 by Karisto, Dora and Mikaberidze, which reported measurement of infection efficiency of a major wheat pathogen using time-resolved imaging of disease progress. So a major wheat pathogen is in fact Zymoseptoria tritici, a fungal pathogen of wheat that causes septoria tritici blotch, which is one of the most damaging diseases of wheat worldwide. And here in the photographs you can see some of the typical symptoms of septoria tritici blotch. So these are these yellow to brown lesions, necrotic lesions, and within the lesions you can see those black round structures. These are pycnidia, where the pathogen produces its spores. And so as you can see, the disease affects primarily wheat leaves. Now, what is infection efficiency and why would we want to measure it? So when a fungal spore lands on a susceptible leaf tissue, it has a probability to germinate, then to penetrate the tissue and eventually form a lesion. And so this probability that once it has landed, that it will eventually form a lesion is called infection efficiency. And it is an important component of reproductive fitness of plant pathogens, and it greatly contributes to epidemic development. And so it is crucial to be able to measure infection efficiency in an accurate manner. And this is what we did in this paper. So we devised this original experimental setup in a glass house under controlled environmental conditions, where we had those metal plates on which we attached those leaves with elastic strings. These were second leaves of young wheat seedlings. And then we inoculated a spore suspension of Zymoseptoria tritici, so sprayed this liquid spore suspension on top of the leaves. So we infected all these leaves. And then what we did was to measure disease progress in each of those leaves using time-resolved imaging, essentially time-lapse photography. So we had resolution in both time and space. And this is what we observed. So the first lesions started to form about 12 days post-inoculation, 12 DPI. And eventually different lesions formed at different locations across the leaf. And pycnidia, those black fruiting bodies, started to form within the lesions. And then eventually the lesions started to merge together. And at the end, the entire leaf has become covered by lesions with pycnidia. And so it is only by doing time-resolved measurements that we were able to count accurately the numbers of lesions on each of those leaves. And then we related the lesion density on leaves to the density of spores that we initially applied in the inoculation. So from the slope of this dependency, we were able to estimate the infection efficiency. So in this case, the infection efficiency was about 4%. So this means that out of 100 spores landing on a leaf, only 4 would cause lesions. So this is a relatively low number, but it is consistent with estimates of infection efficiency from for other pathogens that we can find in the literature. And so now with this methodology, we can estimate infection efficiency for a number of different pathogen strains in a number of different cultivars of wheat, because as I said, from the perspective of the pathogen, infection efficiency represents an important component of reproductive fitness of the pathogen and contributes greatly to epidemic development. But from the perspective of the host plant, infection efficiency, so some hosts can suppress infection efficiency, and this can serve as an important component of quantitative resistance to disease. And so with this technique, we can now estimate infection efficiency across different combinations, across a large number of different genotypes of the pathogen and a large number of different genotypes of the host. And then using the genetic data, the genomic markers in the pathogen and in the host, we can conduct a genome-wide association study or quantitative trait loci mapping in order to reveal the genetic basis of this trait. And this can boost breeding for disease resistance and increase our capacity to control this disease, which is quite challenging. So this is all. I hope you enjoyed this video, and if you have any questions or comments, feel free to leave a comment below this video or just do send me an email.